Good morning, everyone. We'll probably get started in 3 to 5 minutes. Just want to give people a chance to still trickle in. All right, looks like people are still trickling in a bit, so we're going to give another 2, 3 minutes before we get started. Okay, I think we'll slowly get started with this week's Q&A session. So just to begin, I am Andrew Anfora. I am the Senior FAS Manager for Universal Sequencing. Also on with me to field questions is Tom Chen, our CSO. So just if you have not, if this is your first time attending, just like to let you know how we've been running these so far. I'm going to spend maybe the first 5 minutes or so, just giving an overview of our technology, just so we're all on the same page if you're completely new to the technology. Then we will open it up for Q&A. Some weeks we have a little bit more of a structured slide presentation for new applications. I think we're going to do that next week, but this week will primarily be question and answer. You can type questions into the chat at any time. Once I'm done with the slide overview, we can open up the audio as well. We've been having the initial presentation go with the audio muted just so I can get through the overview, and then we'll get to questions. So thank you for attending. If you've attended in the past, you'll notice we have a new picture of the new facility taken on a slightly sunnier day. Universal Sequencing is a bicoastal company. We have offices in Boston, and our primary manufacturing and R&D site is in Carlsbad, California, in San Diego County. So, what is Universal Sequencing's TELSeq? Very briefly, it is a one-tube library prep kit that makes a Illumina sequencer give long read like data, uh, getting you molecular information up to and greater than 100 kb in length molecules if that's in your library prep. Additionally, the library prep requires a very low input, so sub-nanogram to nanogram quantities of DNA. And there's a whole bunch of things you can do with our assay. I'm not going to go through the specific examples, but if there are questions about specific applications, I do have slides covering some of them for show and tell. Really, I just wanted to give an overview of the technology so everybody is on the same page, and then open it up for question and answer. So this diagram should be 2/3 review for most people in attendance, but I'd like to go through it. Uh, it is a depiction of next-gen sequencing platforms. And we're looking at it in the context of a diploid genome. So the first example of short reads is Illumina sequencing, which pretty much everybody's familiar with. So by that, we're talking about highly accurate, fairly low cost per base pair uh, data acquisition for 100 to 500 base pair inserts of unlinked libraries. So you can generate a lot of data using short reads, of course. Two major challenges with the default short read workflow is that it's quite challenging to link haplotypes across a given chromosome. As depicted in the cartoon, you can see the library fragments are too short to capture widely spaced haplotypes. Additionally, it is challenging to get a good genome assembly, a good de novo assembly using short reads due to the short length, challenging to span repetitive regions, end up with a lot of gaps in your assembly and short contigs. So moving down the diagram, there are, of course, true read platforms, which many of you are also probably aware of. Primarily, these are PacBio and Oxford Nanopore. And these platforms can generate longer reads, most typically in the 10 to 20 kb range. And these longer reads are able to more robustly link uh, haplotypes and phase chromosomes than short reads are able to do. And also it becomes technically feasible to do a de novo genome assembly for novel organisms. In the case of PacBio especially, you do have the option of highly accurate uh, moderate length or less accurate, slightly longer read lengths. So there do remain challenges with long read platforms in terms of both base pair calling accuracy and or cost per base pair. PacBio being a great example of you can choose either accuracy or lower cost per base pair, not both. So the solution that Universal Sequencing has created to really bridge the limitations of both technologies, is a linked read product called TelSeq. I'll get into the mechanism of how our library prep works in the next slide. Just at the high cartoon level, what we do is we apply UMIs to the intact DNA pre-fragmentation, and that allows us to trace back the short read library fragments to a molecule of origin. And we are not constrained by molecule length. So unlike PacBio per se, where you can get highly accurate reads up to that 15, 20 kb length, our only constraint is the DNA length, molecule length going into library prep. So it's very routine for us to capture molecules uh, greater than 100 kb in our data sets, as long as you're isolating DNA of that length. So that super long length really lets us do a lot of things that you can't even do with some long read platforms. I want to somewhat briefly go through this and can circle back if there are more detailed questions, but our library prep is fairly straightforward. I like to point out that everything takes place in PCR tubes. So if you're already running next-gen sequencing library prep, you likely have all of the equipment needed. There isn't any sort of exotic assay-specific equipment that's required, just all standard molecular biology tools. So what we do is the first step in our assay, and we're going to be working left to right on this diagram, is you take your, you will be mixing a transpososome complex with our TEL beads. The TEL beads are beads with an 18 base pair barcode that is bead specific. So we have a potential library of 2 billion barcodes. So we are able to handle large genomes and really capture a lot of diversity from our large number of indices. We will then take that bead and transpososome mixture, add in very long DNA. And I say very long DNA. Ideally, we want to be using DNA that is greater than 40 kb in length. It is possible to use DNA shorter than 40 kb. Really, you're going to start to see diminishing returns with the advantages of our assay if you can't get DNA at least 10 kb in length. So you'll take your long DNA, you'll take your TEL bead, take that first transpososome complex, mix in a tube. That will then form a complex of transpososome binding the barcode and binding the DNA molecule. We then add a second transpososome for tagmentation. Uh, that will also bind the super long DNA fragments. We break and wash the DNA, and we will generate our libraries by amplifying DNA with the subset of the DNA with the proper orientation of transpososome complexes. And our PCR will leave you with a library containing Illumina compatible P7 and P5 adapters. Again, really straightforward. Total turnaround time for our assay from isolated DNA to sequencer ready library is 3 hours. So once you're proficient with the assay, it would be possible to isolate your DNA, generate your library, and start collecting sequence data uh, all on the same day. Just briefly, and I don't want to spend too much time on this unless there are questions I need to circle back on, but we have several free software tools currently available. So on this diagram, we see really the two major branches that our customers take. Depicted in green are the universal sequencing provided software tools. So we have a QC and denoising tool called TelRead. That is what you'll run on all data sets coming off of your sequencer before you do anything else. At that point, there is the option of going into a couple of different analyses. We have TELLink, which is our de novo assembly tool, and we have TELSort, which is our phasing tool. So all three of these pieces of software are freely available on the Universal Sequencing website. You can get them as either Docker or Singularity images. And TELRead and TELLink are currently available on BaseSpace, and TELSort is in the final stages of beta testing on BaseSpace as well. So if you just prefer running your everything through BaseSpace, that can be done with our software. It's really easy to do. I'm glad we have it up on BaseSpace. Additionally, our data type are quite similar to the legacy 10x Genomics linked read data. So we do provide a conversion tool that converts our 18 base pair barcode into a 16 base pair barcode compatible with 10x's pipeline. So you can use Long Ranger or Supernova if you're already familiar and like those programs. Additionally, you can take the cleaned up FASTQs out of TELRead and do something else, of course. And really that takes me to just, again, the summary, quick workflow, low input, with very long DNA molecules preserved, and our software is freely available. So I will pause for questions. If I'm seeing the right chat window, I don't see any questions yet, but feel free to type in questions. David, if we're not getting questions typed in, could always go ahead and unmute people.
I did go ahead and allow people to use the mic. So if you would like to unmute yourself to ask a question, please go ahead and do so. You can also type questions in the chat, of course. Looks like we have one coming in. Just
Yeah.
one.
Yeah, I figured out how to keep both windows open, so I can see them too. See, two questions being typed. And we'll be emailing a little bit more about it. Just for those of you that were hoping for a little more of a structured presentation, what we're probably going to do moving forward is 1 week a little more open-ended and overview-ish, and every other week do a little bit more of an application deep dive. So we'll be emailing more information about what next week's presentation will cover. Oh, okay, great. So first question that came through in the chat, there are enough reads to assemble a mammalian genome from one reaction. Yeah, so our kits contain enough reagent for up to 4 human sized library preps in our lower throughput kit. And that will give you enough library for sequencing a human-sized genome. Definitely enough library. Yeah. So, and by that I mean typical mammalian-sized genome. If it's something like crazy, like a salamander genome, not sure what the largest genome we've done with a single library prep reaction, but in general, get enough library out that's not going to be limiting. Okay. Another good question. Is it okay to mix TELSeq with other types of libraries on a flow cell? So our Our libraries do require a spiked-in sequencing primer, but the spiked-in sequencing primer does not cross-react with the standard Illumina sequencing primers. So our samples can be run on the same flow cell as a standard Illumina library with no problem. A good question there, and the data can be demultiplexed fine. There isn't cross reactivity with standard Illumina sequencing primers. If for some reason you're running a mixed flow cell with a secondary spiked in non-standard primer, we'd be happy to review the primer sequence and confirm that it's not cross-reactive. We haven't yet seen other sequencing primers that cross-react with our libraries.
And again, just a reminder, you guys are welcome to unmute yourselves and ask your question directly as well as typing in the chat. Andrew, did you cover at all what our typical bundles and packages cost?
No, I didn't. I don't know if I have that in this set of slides. Let me stop sharing and see if I have that, if you want me to pull that up. One sec. Might need to jump to a different deck. Yeah, I don't have it in this one. David, do you want to share that, or do you want me to? I can put that up if you want.
So I'll just share it verbally. Our standard bundle, which is typically enough for preparing 4 genomes of 1 GP or greater, that is $900, and our high throughput kit, which is enough for 24 libraries, is $4,490. And Andrew, you can expand on those kits a little more if you'd like, in terms of what they can do.
Yeah, so our standard volume kit is enough reactions for 4 human-sized genomes or 12 microbial genomes or some intermediate number of intermediate-sized genomes. Our protocol goes through 4 different genome bin sizes that with suggested reaction volumes and reaction conditions. And our higher throughput kits can process 6 times the number of samples, so 72 microbial samples or 24 human-sized genome samples.
What is the smallest? So the question is, will any genome be too small? So I think it's fundamentally, what's the smallest genome TELC can process?
So there shouldn't be a lower limit to genome size. And I think next week's presentation will get into some data. We are bringing online a Amplicon protocol. Right now, our currently launched protocols are focused on whole genome. We do have a hybrid capture protocol for working with Agilent SureSelect, and we are rolling out, going to be rolling out some information about Amplicon-based linked reads as well. So really, in terms of genome size, nothing is too small. Depending on the question that you're trying to ask with your research. Really, the limitation, the one limitation would be in there. I'm going to stop sharing and jump to a different slide. That is a good question. The one limitation with our assay, not even limitation, but the one concern with our assay is going to be the length of DNA that you're able to isolate is going to determine how contiguous your assemblies or phase blocks are going to be. So when I said that we want DNA that's 40 kb in length or greater, it's really from studies like these that we generated those recommendations. So what you can see on this graph is the relationship between gDNA molecule length and observed phase block for phasing a diploid genome. So really, that's going to be more of a limitation, a constraint on your resultant data than the genome size. So as long as you're getting DNA greater than 10 kb for a whole genome application, then really the genome size doesn't matter too much. And we can also do smaller targets that would have slightly different considerations that we'll be talking about a little more in the future.
All right, any more questions from anyone in chat? Doesn't look like it. Okay, well, I guess we can go ahead and start wrapping up. Once again, thank you everyone for attending, and we look forward to seeing you next week. Thank you so much, Andrew and Tom, and everyone else for attending. Oh, it looks like we might have one more coming in real quick.
Thank you. Yeah. Thank you, Andrew.
Yeah, my pleasure as always.
And again, if you do have any further questions, feel free to email us here at Universal Sequencing. You should have our direct emails. And once again, thank you. Enjoy the rest of your day.
Thank you. Bye-bye.